Hello, I'm Doug Clark from the University of California at Berkeley, and it's my pleasure to welcome you as the Editor-in-Chief of Biotechnology and Bioengineering and to introduce the 2020 B&B Daniel I.C. Wang Award. This award was established over 10 years ago and is named after a very esteemed and beloved biochemical engineer. Danny Wang's contributions to the field of biochemical engineering are too numerous to recount here and now. However, the special issue of B&B commemorating Danny's 70th birthday contains many articles that describe his enormous impact on the subject matter and practitioners of biochemical engineering. Danny was also editor-in-chief of B&B from 1984 to 1990 and has published over 100 articles in B&B alone, spanning the range of topics shown here. And as a longtime admirer of Danny once said of the man and his career, from basic research to bioprocess development, from educational mentorship to industrial leadership, and from local administration to international statesmanship, Danny has enriched the landscape of biotechnology at all levels. Therefore, there is no one who better personifies the qualities of this award, which is presented to a researcher under the age of 35 or under 8 years from being granted his or her PhD, to recognize exceptional research contributions and sustained commitment to biotechnology and bioengineering. And I'm very pleased to announce that the recipient of this year's B&B Daniel I.C. Wang Award is Peng Xu, Assistant Professor, Department of Chemical, Biochemical, and Environmental Engineering, University of Maryland, Baltimore County. Peng received his BS and MS degrees in biochemical engineering from Jianan University, and then went on to obtain his PhD in chemical engineering from RPI, working with Mateos Kafas, where his contributions included developing a versatile gene assembly and cloning platform, which he called ePath Brick, to solve the challenge of assembling multiple gene pathways and fine-tuning gene expression. Following postdoctoral work with Greg Stephanopoulos at MIT, where he made several important contributions toward developing yeast-based biorefineries for producing oleochemicals, Peng joined the faculty in the Department of Chemical, Biochemical, and Environmental Engineering at UMBC in 2016. Peng's current research focuses on metabolic engineering, synthetic biology, and biochemical engineering. His laboratory is perhaps best known for integrating genetically encoded biosensors to control and reprogram cell metabolism for diverse applications. Peng has also expanded his work to apply feedback control theory and computational modeling to improve the robustness of microbial cell factories. And here is what one of Peng's references for the Wang Award had to say about him. Quote, Peng is an excellent scientist and engineer who knows how to apply basic scientific principles to solve real-world challenges. His enthusiasm will train the next generation of students, and his technical skills and insights will benefit us to develop sustainable biorefinery platforms and engineer biosensors. His concept on artificial intelligence and dynamic control will surely benefit the entire synbio and biomolecular engineering community, end quote. Besides winning the Wang Award, Peng has received numerous other honors and accolades, including a Bill and Melinda Gates Award for novel artemisinin manufacturing technology and serving as an editorial board member for the journal Metabolic Engineering. Which brings us back to the Wang Award, and Peng's B&B portfolio is shown here. And I should mention that the first paper on this list garnered Greg Stephanopoulos the Elmer Gaydon Award last year. So Peng is the only co-author I'm aware of whose efforts have brought home both a Gaydon Award and a Wang Award. Now, Peng obviously doesn't publish only in B&B, although we wish he did, but it's clear from this work alone that he is quickly establishing himself as one of the young faculty stars in the areas of synthetic biology and metabolic engineering. And so, Peng, this is our way of acknowledging your burgeoning leadership and your excellent work and saying thank you. Now it is time for us to welcome and hear from Peng Xu.
First, I would like to thank Doug for the kind introduction. I hope you are all doing well, and the virtual meeting will run great today. I would also like to thank the organizing committee of the ACS meeting, especially Mark and Brian. They are working very hard to put the ACS BioT meeting on our schedule. Today, it's truly a great honor for me to present the BNB Danny Wang Award lecture. I'll start with my connection with Danny. Danny's life is full of memory and achievements. We all knew his legacy and long-term commitment to the BNB community. I just want to mention a few things that my footprint came across Danny's life, which tremendously benefited my career. Danny was born in Nanjing, 1936, just 1 year before the Sino-Japan War. His parents came from Suzhou. I did my undergrad college at Jiangnan University in the early 2000s in the city of Wuxi. So right in the middle between Nanjing and Suzhou, either direction, it's about 1 hour's train, 50 to 70 miles away. During my college, we had a large collection of English textbooks. One of the textbooks we used is Danny's work, Fermentation and Enzyme Technology. Suzhou, Shanghai, Wuxi has a tradition to make fermented food in terms of quality and variety. Somehow, I believe this culture fostered a young man of Danny. That's why he decided to pursue a degree in fermentation and food biotechnology. He managed to come to the US in 1950s and completed a degree in MIT and UPenn. Later on, I joined Stephanopoulos' lab as a postdoc. I used the plate readers, microscopy, and bioreactors on a daily basis at the BPEC Center. Danny was the founder of the BPEC at MIT. The center is still running today. In 2015, I was fortunate to work as a teaching assistant for the week-long professional course, Fermentation Technology. Danny is the director and founder for this course. At that time, Dr. Kristala Presser was the primary instructor. The course has been running for more than half a century and has trained thousands of young professionals who became leaders in the pharmaceutical industry. The most memorable thing is on the graduation day, Danny was sitting on a wheelchair and will greet all the students. I couldn't forget the big lobster they serve on the graduation day. Suffice to say, Danny's legacy and commitment is always an inspiration for me to work harder. Before I start the lecture, I would like to express my gratitude and sincere thanks to all the people who came across my career. This award lecture is not just for me, it's truly a celebration for my mentors, my colleagues, my collaborators, my friends, my students, and of course, my family members for their consistent support. I won't be able to name them one by one. I just want to emphasize a few of them, including my PhD advisor Matthew Kofas at RPI, my postdoc advisor Gregory Stephanopoulos, and Wilfred Chen at Delaware and Yinjie Tang at St. Louis, who nominated me for this award. At last, my thanks go to BNB and Valley for supporting this award. For string engineering, we routinely found there are two major bottlenecks. The first one is competing pathway. We can generally knock out the byproduct secretion pathway. But some of the essential byproducts, let's say fatty acid or lipids, which are essential to cell growth, we cannot simply knock out that pathway. What we can do is that we can build a monitoring and regulatory system in the cell. Once the byproduct reaches the critical amounts, the cell can automatically shut down gene expression of this pathway. We name it negative autoregulation or inverter gate, so the cell always maintains gene expression at the right level. The second bottleneck is the long-term strain stability. With cell passage at large-scale fermentation, we routinely observe some of engineered cells will lose the production phenotype and degenerate. As the cell passes from one generation to another generation. At the population level, the question is how we can stabilize long-term strain stability. The solution here is a growth-coupled reaction. We can link our target compound with an essential nutrient or cofactor that are incredibly important to cell growth. The cell will be metabolically addicted to this intermediate, so we can force the cell to maintain both growth fitness and production. A couple years ago, there is an excellent work published by Tom Ellis' group, where they have built a burden-driven feedback control circuit to combat protein expression burden. Let's say you have a synthetic construct generating lots of protein expression burden. Its burden signal can be sensed by the HTP-G1 promoter. They use the HTP-G1 promoter to drive expression of the D-Cas9 and GAD RNA. Then the D-Cas9 GAD RNA complex will instead down-regulate the transcriptional activity of the PBAT promoter and reduce the protein expression burden. So this negative autoregulation turns out to be very successful to monitor the protein expression burden and improve the protein productivity. There have been another two excellent examples published by Fuzhong Zhang's group at WashU and Morton Summers' group at DTU. They have separately engineered feedback-controlled genetic circuits that can be engineered or linked to cell growth fitness. For example, they have engineered FADA repressor-based leucine addiction, which is on the left panel, and LC activator-based tetrahydrofolate addiction on the right panel, to respectively improve fatty acid and MOA production in E. coli. They found this metabolic addiction could stabilize long-term strain performance and reduce the non-genetic cell-to-cell variations. So the basic principles are that the leucine or tetrahydrofolate will confer a competitive growth advantage to the engineered cell line. The cell was forced to overproduce the target compound to maintain the growth fitness. A couple years ago, my group started to engineer the flavonoid pathway in Yarra Valley Polatica. We found there are two problems. This yeast makes lots of lipids. We knew lipids or fatty acids compete with the flavonoid pathway for the same precursor melanoic-CoA. Another problem is the long-term strain stability. We routinely observed decreasing flavonoid production when we passage the cell. The first problem is the competing pathway, and the second one is the long-term strain stability. To solve both problems, we have designed a bi-layer dynamic control system by combining negative autoregulation and metabolic addiction. We hope to improve both the flavonoid production and strain stability. I'll just show you how we implement this strategy in a minute. The first thing we do is that we construct the POR1-based fatty acid monitoring system. We use a number of minimal POX2 promoters and engineer the POR1P binding site A1, A2, A3, and R1 with luciferase as a reporter gene. Eventually, we found a synthetic promoter which demonstrated about 20-fold change of transcriptional activity and response to oleic acid up to 5 grams per liter. Then we started implementing the CRISPR-based negative autoregulation to repress fatty acid synthesis. We targeted 3 genes, FS1, FS2, and FABD. Each of them we designed 3 guide RNA targeting sites. We tested the mRNA transcripts of the target gene with qPCR. We found these CRISPR targets could repress gene expression, decrease the lipid content, and increase flavonoid yield. Each of the 3 targets show different repressing effects. It suggests to us this inverter gate could effectively redirect melanoic-CoA from lipogenesis to flavonoid production. To improve the CRISPR repressing efficiency, we combined all the 3 top targets and tested the combinatory effects. We did find there's a negative correlation between fatty acid content and flavonoid titer in our cell, which tells us that the cell could automatically control fatty acid synthesis, and we managed to move some of the carbon flux from lipids to flavonoids. We next move to solve the strain stability issue. Here we would like to build a regulatory system that could sense flavonoids and drive the expression of an essential nutrient or cofactor that is coupled with cell growth. Most of the flavonoid sensing proteins were discovered in prokaryotes like bacteria. Yeast transcriptional bubble is much more complex than the bacterial system. They are consisting of distal control elements, proximal control elements, Tata box binding protein, DNA binding protein, and lots of activator binding sites, etc. In order to eliminate crosstalk between native transcriptional factors and the flavonoid sensing regulator FDR, we tested a number of truncated minimal promoters. We placed the FDR binding sites, FDO, the operator, right in front of the minimal promoters. Eventually, we engineered 3 synthetic promoters that could sensitively respond to naringenin up to 100 mg per liter. Here, naringenin is a specific type of flavonoids. Our next task is to log flavonoid production with cell growth through an essential metabolite. So leucine is a commonly used orthotrophic marker and has no crosstalk with the flavonoid pathway. That's why we choose leucine as the linker molecule. By using flavonoid sensing regulatory system FDR to control leucine production, we did find we can achieve a tight control over cell growth in the absence versus the presence of naringenin, as shown here in the agar plates. The secret here is that we must use the promoter without leaky expression to control the leucine biosynthetic gene. In the liquid culture, we could reproduce this naridinin-coupled growth phenotype. Only in the presence of flavonoids, the cell could grow rapidly. Essentially here, the growth finish is controlled by the amount of flavonoids present to the cell. Its growth-coupled metabolism will allow us to selectively encourage the growth of the cell with increased flavonoid titer. At the end, we combine both metabolic addiction with negative autoregulation to test whether we can improve the strain stability and increase the pathway yield. Shown here, the black square represents the control strain without metabolic addiction. We can see the control strain regained the growth finish but rapidly lost about 95% of our target compound at 300 generation. Instead, our engineered cell could persist about 91% of the target compounds up to 320 cell generations. You can imagine its long-term strain stability or stabilized production phenotype is very important for us to scale up fermentation and shift the economics of industrial bioprocess. In the next 3 to 5 minutes, I'll explore the mechanism how metabolic addiction could reshape the landscape of strain degeneration. We all know that the engineered cell line constantly experiencing metabolic stress, which results in retro mutation. They will kick out the plasmid and lose the production phenotype. We want to ask the question, what is the interplay between metabolic stress and strain degeneration? As a chemical engineer, I would like to answer this question with a mathematical model. Imagine we have engineered cell population X1. There is a constant retromutation frequency theta, leading to the formation of the retromutant cell X2. At the same time, only our engineered cell could make the target compounds through a partially growth-coupled pattern, as defined by alpha and beta in the equations. We can define a metabolic coupling coefficient gamma, which describes the beneficial or detrimental effects the target compound imposes to our system, or to the engineered cell. For example here, the specific growth rate of the engineered cell X1 and mu1 here is metabolically coupled with the target compounds through gamma. A negative metabolic coupling coefficient indicates detrimental effects or metabolic stress. On the other hand, a positive metabolic coupling coefficient indicates beneficial effects or metabolic addiction. By doing the mass balance equation in a CSTR, we could list all the mass balance equations for X1, the retromutant X2, our target compounds P, and substrate S. By solving this equation at steady state, we can figure out the relative distribution for both the engineered cell and the retromutant cell. Then we can find the degeneration frequency delta in our system. So delta is defined as the fraction of the retromutant cell relative to the entire cell population. So delta is a function of theta and gamma. As we solve the steady-state solution, we can plot all the solution as a function of the dilution rate d. For example, shown here, figure A is the steady-state solution of the engineered cell line X1 as a function of the dilution rate. As we change the metabolic coupling coefficient from a negative value to a positive value, which means we change from a detrimental effect to a beneficial effect, or equivalent to say we change from a metabolic stress condition to a metabolic addiction condition. We can see the engineered cell population X1 will increase substantially, and the retromutant cell X2 was reduced substantially as the gamma shifts from negative value to positive value, which is equivalent from a metabolic stress condition to a metabolic addiction condition. At the same time, the target compounds will increase substantially as we change from a metabolic stress condition to a metabolic addiction condition. How do we explain this data? So the dilution rate here is a kind of a selection pressure. The CSTR will only allow the cell with better growth finish to be retained in the system, and the cell with worse growth finish will be washed out or eliminated or maybe unselected from the system. As we dig into the data, we can analyze the degeneration frequency. The degeneration frequency is defined by the fraction of the retromutant cell relative to the entire cell population. For example here, X2 compared to X1 plus X2. Shown here on the left panel, as we change the metabolic coupling coefficient from a negative value to a positive value, which means we change from a metabolic stress condition to a metabolic addiction condition, we can substantially reduce the degeneration frequency. For example here, 10% or 20% of metabolic addiction is sufficient to suppress degeneration frequency under 4%. At the same time, we found a negative metabolic coupling coefficient corresponding to metabolic stress condition, which can accelerate degeneration. On the right panel, we could also see that a 10% or 20% metabolic stress is sufficient to decrease the target compound by 80% or 90%. So take-home message here is that we should always try to avoid the metabolic stress condition and encourage a growth-coupled metabolic addiction condition in strain engineering. In summary, we can use genetically engineered metabolic circuits to solve problems in strain engineering. For example here, we can use negative autoregulation to redirect carbon flux from the competing pathway to our target pathway. When the competing pathway is an essential metabolic reaction that we cannot simply knock them out. At the same time, we can use growth-coupled metabolic addiction to stabilize the long-term strain performance to minimize strain degeneration. The competitive growth advantage engineered in the cell is kind of an evolution strategy to combat retromutation. We also found that the mathematical model is very important for us to understand metabolite-host interaction. We proposed the metabolic coupling coefficient, which allows us to quantitatively explore the solution of metabolic stress or metabolic addiction strategy in strain engineering. In the end, I hope all the best for the ACS community, especially my best wishes go to Danny and his family. It's truly my great pleasure to present this lecture, and I will be happy to answer your question later.